All right, good morning. We are right on time. Perfect. All right, so welcome to Introduction to Ecology. More importantly, I guess, welcome to second year. Universally accepted as the best year of your university education. You've sort of gone through that weird introductory first year phase. You're not quite worried about finals and things like that yet. So second year and third year are, certainly in my experience anyway, the best time to be a university student. And one of the really nice things about this time is that you start to move a little bit towards specializing your courses and specializing your classes into topics that you're really interested in. And we're going to talk a bit about, in this course, a bit about ecology, and I guess you did certainly would have touched on aspects of ecology in the introductory biology lectures last year, biology courses last year. We're going to go a little bit more detailed into some of the more interesting, at least I think, some of the more interesting aspects. Start at the start. So who am I? That's me up there, if you haven't guessed. My name's Brian Hayden. I'm a research associate here at UMB, which basically means I'm part of the biology faculty, but I mostly do research and I do a bit of teaching as well. I run the SynLab, so the SynLab Stable Isotopes in Nature Laboratory, which is a ecological lab. We use a specific tool that allows us, basically looking at an organism's chemistry, to identify what it's eaten or where it's been or things like that. And I use this tool to study aquatic ecology. I'm mostly interested in, let's see, will this work? No? Okay, we'll survive. I'm mostly interested in fish, also some aquatic invertebrates in lakes and rivers. Initially around New Brunswick, but also across the globe. So this is a quick slide that sort of shows some of the research projects that I'm involved in. And the goal of this slide is sort of to show to you that this is what ecology can be. This is what ecology is. We're doing the same type of research across the globe. Here in New Brunswick, I'm going to go old school and use this super pointy stick. Up here, I've got some projects looking at Atlantic salmon. Anybody know what they are right at the top? Lamprey. Who said lamprey? Super extra bonus points. So lamprey. Lamprey are kind of like eels, much cooler. And Canadian lynx. We'll talk a little bit more about all of those at various different phases during the course. But in addition, we've got using stable isotopes, using this tool, and studying trophic interactions with birds, marine fishes, other birds, fishes in Europe. I'm from Ireland over here. Well, that's Iceland, but I'm from Ireland over here, and I've been in New Brunswick for around about 4 years doing this kind of work. That's what I do in ecology. But ecology is a really broad discipline. And what we're going to study or what we're going to learn in this course is really an introduction to this discipline, an introduction to what exactly we mean by ecology. We've got a series of sort of learning outcomes, and these aren't dictated anywhere. These are what I want you to come away from this course with, and what I hope you want to come away from this course with. And initially, you've got an understanding what is ecology. When we talk about ecology, exactly what do we mean? What different types of ecology are there? Different sub-facets within the discipline. Why is it important that we know this information? What can we gain by a knowledge of ecology? And what type of insights about the natural world can we gain by knowing the ecology of the organisms we're studying? Okay, some basic housekeeping stuff first. There's 2 recommended textbooks for this course. Our main focus, a lot of the examples I talk about in class will come from this up here, Fundamental Elements of Ecology. This is initially this version is quite an old version. We're doing an introductory course. We don't need to have a new book every year. The concepts that we're doing here won't have changed massively. So if you've got this version, or if you can get a slightly different version, either of those will do. This will be available. I've got a couple of copies. There will be a couple of copies on reserve in the library. There'll also be copies available from students who took the class in other years. We've used the same book. I've taught this course for 3 years, and we used it for a few years before that. So there are quite a few copies floating around that won't cost you $200 to buy. I'm going to talk a bit for a couple of my lectures about analysis of ecological data and how we analyze data in ecology and why it's important to be able to do that and know what we gain from that. I'll use examples from this book here, The Analysis of Biological Data. And again, there'll be a copy of that on reserve in the library. The course is co-taught between myself and Professor Stephen Peake, a professor here in the biology department. You'll have me, where are we, up until the middle of October, and then Professor Peake will take it on till the end of term. You can sort of see what I'm going to talk about. Like I said, we have some introductory statistics, and we talk about climate, physical environments, the abiotic components that sort of determine what type of species live in an area, how the abiotic components influence the biotic components of the system. At the end of my section, we'll have a midterm exam. At the end of Professor Peake's section, there'll also be a midterm exam. These will be multiple choice questions. The final exam will also be multiple choice questions. Each midterm exam, I think we decided will count for 20%. So by the time you've got the two midterm exams out of the way, you could already have 40% of your final grade for the course. You will. All that information is in the syllabus, which is in D2L. You've already all been here for a year already. You know, I certainly hope you know what D2L is, how to access it, go in and get that information. Just this morning, I set up some additional information for the course with lecture notes, small scale ones if you want to print them out, large scale ones if you want to look at them on your laptop while we go through the course. I'm also trying a thing where I'm going to try and record the lectures as I speak, as I do, and provided that works, we'll put a link to YouTube or whatever up into that D2L as well, so you can follow back on what I've said if you've missed anything. While the course will work from the prepared notes and examples from the texts, I'll also refer to primary literature sometimes, to actual research papers or general books of accessible scientific interest. And where possible, I'll put those specific papers or books into those units as well for each lecture. So all the information will be there and available if you want to get to it. Any thoughts, questions? Clear? Excellent. If you do have any questions, feel free to just stick up your hand or shout out. So I'm happy for this to be a discussion as much as I can sit here and talk at you every 3 hours a week until the middle of October. I can do that, or we can have a discussion. What works? What we're going to do in this class is sort of take a first little look at start addressing some of the first questions that we have for the course. Initially, I want to tackle that one question: what is ecology? What do I mean by ecology? How broad is it? How do we subdivide it down into different disciplines? Then the next section is sort of how do we do ecology? What is, and to do ecology, like all science, we use the scientific method. but how do we apply that to an ecological setting? And then we'll have a little bit of talks, or a little bit of work, or a little bit of discussion about some of the ecological work that's going on here in UNB, and the opportunities for students, for young undergraduate students who are interested in getting more hands-on experience in ecology. We are in a wonderful place to study ecology. It's one of the reasons I came here 4 years ago, and still quite happily here. But we have migratory birds, and there's some bird work that's going on in the Bay of Fundy, unique populations of migratory birds, the actual Bay of Fundy itself, which is just a wonderful place to test hypotheses and to study and to learn about marine ecology. All sorts of interesting freshwater ecosystems with all sorts of interesting freshwater phenomena going on. We'll talk a little bit about that as we get towards the end. So, what is ecology? In its broadest sense, ecology is the study of the interactions between organisms and their environments. That includes their interactions with other individuals, with other organisms, and also with their surroundings. So this is a plus. So I'm doing a lot of my research. I mentioned I do sort of aquatic ecology. A lot of the work I do relates to food webs and understanding the trophic links within food webs. And if you want to know about humpback whales in the Bay of Fundy, you need to know about everything else. As far as I'm concerned, you need to know about everything else that feeds into a system which can support a humpback whale. So in this example, we've got a marine environment with near shore or a benthic type component, an offshore pelagic type component. In different habitats, we have different types of species, different types of consumers. Down here, species which are adapted, such as shells, filter feeding clams or oysters, are adapted to being a sessile benthic organism in the open ocean. They can maintain the rigid structure to the benthic, to the benthos, to whatever structure underneath them, but at the same time, they're filter feeding pelagic phytoplankton, particulate matter from the water column. They're fed on by different sort of benthic consumers. As we go higher in the ocean, we get into a photic zone where light can penetrate. Light stimulates plankton growth. Phytoplankton, tiny microscopic plants, are fed on by not quite so tiny, almost microscopic plankton crustaceans. They're fed on by slightly bigger crustaceans. They're fed on by smaller fish. They're fed on by bigger fish. And eventually we've got a system which can support whales. Ecology is not environmentalist. Ecology is the work, so people, certainly my generation, are familiar with Greenpeace. People of the younger generations are possibly more familiar with things like Sea Shepherd, organizations which actively pursue conservation, actively pursue a particular conservation agenda. That can be a valuable service, but it's not ecology. Ecology gives us the information which can allow us to understand how we achieve environmental goals, how we can institute a successful conservation program. Which types of a population do we need to manage or do we need to conserve? Do we need to ban all whaling? Or can we identify specific populations which are threatened, specific populations which are not threatened, and understand how better to manage the system? Well, not manage the system, but mitigate our impact on the system. That's what we're studying in ecology. I think of it as it's the mechanisms through which the environment operates. It's incredibly broad and diverse. It's an incredibly complex science. As you'll see over the course of over this lecture, or this series of lectures, and this is just the tip of an iceberg. This is just an introduction to the discipline, to the field. I mentioned a little bit earlier in some of my ramblings about abiotic and biotic components of an environment. Biotic essentially means living component, abiotic is non-living. But if we want to understand any habitat or any ecosystem, we need to integrate both of those parts. We have to understand that both of those are playing a part in the dynamics which support this ecosystem. So let's take a forest. We want to understand, we want to conserve this particular patch of, let's call it first generation forest, prime forest. There's an abiotic component to this that's got nothing to do with the plants that are living there, or an issue. That's the climate, how hot it is, how warm it, or how hot it is, how much moisture is available, the soil, or the type of rocks and minerals that are available through the soils. As we'll see later on in subsequent lectures, those three things determine what type of plants live in an area. And what type of plants live in an area determine what type of animals live in an area. So already, the abiotic component is determining, you can say it determines what the biotic component is. But the biotic component can also feed back into the abiotic component. Once we have a lot of plant growth in an area, certainly in somewhere like New Brunswick, where we've got a lot of deciduous trees, we get a lot of dead biotic material, leaf litter, falls down onto the soil. Those nutrients are absorbed into the soil. The soil can then becomes essentially more fertile, and it's suitable for different types of plants. Similarly, if we have a lot of, or a big biomass of life, of photosynthetic organisms there, that can change the oxygen availability. We think of ourselves in an aquatic environment. It can change the actual temperature. We can get microhabitats and microclimates determined by the biotic component. So these two facets are constantly interacting. But if we go and study this area now, we'll see something that looks like that. If we go and study this same area in January or February, we're dealing with an entirely different environment, solely due to abiotic components. But the biotic components are influenced by that. To understand this is an infinitesimally large and complex problem to try and solve, and to understand each facet of what determines the type of ecosystem that can develop in an area and how we can better conserve or manage that. And it involves an understanding of biology, chemistry. The work I do, I was a hopeless chemist in my undergraduate years. I did it the first year, failed horribly, and I now use chemistry to understand biology. I now look at the chemical makeup of an organism and use that to try and infer what it's been feeding, infer its ecology. We can't do that. These two things all of a sudden are integrally linked. Chemistry at its core is physics. And like I said, if we want to also understand the abiotic component, we'll see later on how the actual geology of the soil is a key facet of that. that's a broad picture. And it's very difficult for people, for researchers to tackle that. You can't tackle such a broad picture as a PhD dissertation, or as a master's dissertation, or as an undergraduate project. You can try and do it through a research career, but we need to break it down. We need to identify, or we have identified, specific levels within this overall process, or specific levels at which this overall process occurs. And again, this is taken from your book, but this is sort of standardized across the fields. There's typically 5 levels of ecological organization: an organism, population, community, ecosystem, essentially, and finally landscape. At the organism level, we're dealing with the individual. How does a specific bird react to changes in temperature? Does this specific bird stay in its environment? At what point has its food become so scarce that it needs to move to a different area, that it's excluded from one area? We can look at this, we can then move one up and take a population level, population ecology. The population here is all individuals of a species within one defined space. That's all it is. So we can lock the doors here, and we would be one population of humans, and we could study this one specific population. If we're into sort of the next step up, is to look at the community, at a community ecology. And that's simply the interactions between different populations within one defined space. So a lot of if we go back to the example of understanding whales in the Bay of Fundy, each of those nodes in the food web, each of those species within the food web that channel resources and energy up to whales could be considered, is a distinct population. And we can look at the community by looking at the interactions between the populations. One higher is the ecosystem level. And here we're looking at how the community, we're looking at the community within a region or within a defined space, but we're looking at how that is channeling resources and how that is moving energy through a community. And also how it's influenced or how it's interacting with the abiotic component. From here up, we're primarily talking about biotic. Once we get down to ecosystem, we're starting to integrate some of that abiotic component. And finally, landscape level, so we can take a global level, we're looking at how various different ecosystems interact. We can look at this in New Brunswick. We're interested in how forestry management practices affect the flooding and water levels within the St. John River. We're interested in the forest ecosystem. We're interested in the river ecosystem. We're interested in how the abiotic component, how our management of the forest is influencing how our management of the biotic component of the forest is influencing the abiotic component or the water levels within the system. Similarly, so that's our sort of predefined levels that we can study ecology at. But there's this whole plethora of different disciplines and sub-disciplines within ecology. This is an example of some of the sort of more common ones, but this list could be endless. Conservation ecology, understanding ecology essentially from the viewpoint of trying to conserve a particular species or a species. And this is maybe where we talk about these examples of Sea Shepherd and Greenpeace and groups like that, who have a conservation goal, but we as ecologists need to inform those goals and inform those practices. And we can, when we study ecology from that perspective, it's termed as conservation ecology. Next we have, next on our list at least, is behavioral ecology. How does the behavior, quite often of an actual individual, this occurs at the organism level, how does the behavior of an actual individual respond to changes in its environment? So let's put it next. Physiological ecology, paleoecology, the study of how broad-scale changes all through time. There's a subsequent course, this on specifically on paleoecology here in UMB, and a lot of that work can focus on looking at things like lake sediments. If you drill down through the sediment of a lake and pull out a core, similar to aging tree rings, you can look back through tree rings and count back through time. You can do the same thing in lake sediments or marine sediments. Look back, take a core, 2 meters depth, and look back and you're seeing what type of organisms settled out on the lake bed at that time. And we know a lot about these different organisms, these different plankton, or pollens of specific plants. And from that you can infer what type of plants were growing around the lake at that time, what type of plankton species were living in the lake at that time. Because we know about those species, you can say what temperature, what type of climate was around the lake at that time. So by studying the species that are present in a population or in a site, we can make inferences about the environment around it. And that's largely what we're doing in paleoecology. Biogeography is where we take sort of the broad ecosystem or landscape level concepts we talked about in the previous slide. And here we're sort of saying, okay, I post a paper. It was a research paper that was just published yesterday, and I was going to put a slide in, it didn't. And what they did was they took satellite data, essentially you could think of it as Google Map data, back through time, and looked at the watercolor in lakes across the United States. Watercolor is related to the amount of plankton in the lake, how the amount of excess nutrients coming into the lake, how humic the lake is, the amount of sediment in the lake, things like that. And by taking these global images or landscape level images, they can plot how clear the water, how essentially relate lake water color to human processes. Lakes closer to cities, closer to industrial centres are greener. They've got more phytoplankton, they've got more nutrients in them, stimulating more plankton growth. Lakes in national parks or pristine areas are clearer water. They're nutrient limited. Lakes close to forestry or to large agriculture areas are often quite humid, quite dark coloured because of the extra sediment that's entering the systems. By looking at these patterns, these large-scale patterns, we can make inferences about landscape ecology at a biogeographical level. How do we do this? How do we actually study ecology? Ecology began, or it's a recent, or it's a constantly enriching and developing field. And what we term as ecology really began, or can be traced back to the old Victorian scientists, Victorian naturalists, who went on tours, went on trips, collected samples, documented what was around them. A very descriptive form of science. Went and made observations, didn't necessarily test or try to determine what was behind it, but gave us a rich understanding of the diversity of life. And this is sort of continues to go on to this day, where we have in the spring, I take classes from kindergarten to third or fourth grade in middle school out into rivers and ponds, and we look at the type of invertebrates that are there. It's descriptive. We're not trying to test anything or understand anything. We're just having a look and seeing what's there, developing an understanding of the world around us. But modern ecology, what we're doing now in ecology, is taking that broad descriptive base and trying to tease it apart and trying to understand the mechanisms behind it. Why do we see a massive biodiversity in tropical rainforests? How do species respond in a place like New Brunswick, where we've got often a 60-degree range in air temperature between July and January? This involves applying science to natural history. And this really started in the last century, and it's where, close to where I'm from Ireland. This is Charles Elton, who did phenomenal work in the UK and a number of forests around Oxford University, and was one of the first people to sort of put and develop a mechanistic understanding of why we see different species in different areas at different times. At the same time, we were Odom doing the same type of work, similar type of work in the United States, building what we would now call an ecological network, looking at the species that are present in a particular area at the time and developing a mechanism through which we can explain or predict what species will be where at a specific time, how species are interacting with each other. And this is a big step forward. This is a revolutionary change from simply documenting what's there. And it's applying the rigors of science to natural history. And it allows us to make predictions about what will happen to New Brunswick if the temperature increases by 2 degrees. What will happen to the ocean if the oxygen levels decrease by 5–10%. To do this, we need to apply, or we've got a series of methods, or a series of routes. But essentially, we're using the scientific method to test, to make predictions about the natural world and ultimately to test them. Okay, you should all have heard quite a bit about the scientific method in the prereqs for this course. You should know quite a bit about what I'm going to talk about in the next 10 minutes-ish. But as a refresher, we're going to run through this again, because this is the critical step that brings science and the science of ecology from natural history. So the scientific method starts with an observation. It's a way of making test mechanisms from an observation. We start at the top, make an observation. We've got an example. Run through it with the example. How about we do that? Okay. We're traveling from Toronto to New Brunswick to come to UMB for back to term, because everybody knows that's a better way to travel than west. And on the route, you'd notice that some grasslands appear to be more productive than others. It's an observation. You can document it. It's a classical sort of natural history observation. We then have a question that arises from that, because we've got inquisitive minds. You're coming back to term. Why do we see this? Why are some grasslands more productive than others? A question that's derived from the observation. From that we can pose a hypothesis. A hypothesis is a potential answer to this question. And in this example, we could say that soils with more N here for nitrogen or nutrients allow for better plant growth. Makes sense. You've studied some first-year biology. You know a bit about how plants grow, what plants need to grow. You're traveling through, you're making an observation. This is leading to a question. You're coming up with, you can think of a hypothesis as almost as an educated guess. Yeah? Soils with more nutrients, more nitrogen allow for better plant growth. Critically, this is a question that's posed in the form of cause and effect. More nitrogen, potential cause, allows for better plant growth. From that hypothesis, we can then make a prediction. So let's say a prediction could be that if we add more N, add more nitrogen to a soil, we should see more growth. It's testable. This is something that we can go and design an experiment or make a series of measurements or run a mathematical model to test this prediction. And based on the results of our prediction, we can either say that supports our hypothesis or it refutes our hypothesis. So let's follow this through. Our hypothesis is that soils with more nitrogen allow for better plant growth. From that, you make a prediction that productivity will be correlated with nitrogen across grassland sites. From this, we can design an experiment. We can say, okay, let's get a whole series of grassland sites controlled for the same type of climate, the same amount of light. And in some of these sites, we'll add no nitrogen. In some of them, we'll add a small amount. In some of them, we'll add a lot. We'll replicate this a series of times, allow our experiment to run, measure the productivity, and measure the relationship between nitrogen and plant growth. Thankfully, somebody's already done this for us. So this is in Minnesota, series of different plots, pretty much standardized, all standardized, except for the amount of nitrogen that was added. We can see here, just visually by looking at it, some of these areas have very little plant growth, some of them are very lush plant growth, and everything in between. We're going to talk later on about how we might analyze that statistically, but initially, we can just plot up our data, and we can say, okay, on the x-axis, the amount of nitrogen available, so it could be the amount of nitrogen we added to the soil, the productivity here, which is grams of dry mass per meter squared per year. And we've got certainly what seems to be a very strong positive relationship. So how does this relate to our hypothesis? Does that support or refute the hypothesis that we set forward? Supports it. Does it prove it? No. Very good. Everybody who said no, very good. You can't prove a hypothesis. We do some more tests, you can only disprove it because it's cause and effect. So in our example here, or in this case, so we did our hypothesis. Here we said, okay, yes, in our case, the results of our experiment support the hypothesis. This can lead us to another hypothesis. How does this, is it just nitrogen or is it phosphorus or other elements involved? How does this relate to specific types of plants? The list goes on and on and on. If it doesn't, if you get a no, then you've refuted your hypothesis. In that case, nitrogen does not relate to plant growth. Something else is driving the trends in plant growth that you saw. Design another experiment, test it, rinse, repeat. We can test, quite heavy. We can test this, or we can test hypotheses using experiments. We can also test them using mathematical models. This is a study which is very close to my heart because some of the research I'm doing here in New Brunswick. Interactions between lynx and snowshoe hare. And this is a phenomenal data set developed between 1850 and 1930, which is the number of snowshoe hare and lynx pelts collected by the Hudson Bay Company between those 2 years. And if we assume that trapping effort was standard, we can assume that these are reflective of the population size of those 2 species. So snowshoe hare in red, we can guess that at this point, there were more of them than there were at this point. And one thing we see is that 2 species, the populations of them, almost appear to be in sync. They almost appear when there's a boom in snowshoe hare. It's almost correlated with a boom in Canadian lynx. Here we can sort of think, okay, so what's the relationship between these 2 species? Which species? Is it the abundance of prey or the prevalence of the predator, which is determining this relationship? We think that, let's start with the hare. If there's no predators, the hare population will continue to grow and grow. But the hares are limited by the predators, by the lynx. So the lynx feed on the hares. The hare population will plateau and start to fall as the lynx population grows. Because there's an abundance of hares, there's lots of food for the lynx. They can grow large, have lots of babies. Eventually, they've eaten all the hares. There's no food for the lynx population. The lynx population crashes. There's now no predators for the hares. So the hare population starts to boom. As the hare population increases, times get better for the lynx. The lynx population increases, and the cycle repeats itself. We can model that mathematically in what I'm going to tell you today is the Volterra model. You don't need to understand exactly what's going on here, but what that is essentially mathematically saying that the population size of lynx is determined by the growth rate of lynx, the population growth rate of lynx, and the number of hares. And the population size of lynx is determined by the population growth rate of hares associated with the number of lynx. We can mathematically combine these 2 things together and simulate what we would expect their populations to look like if that was the case, and then compare that to the data we observed. And this is a way that we can test hypotheses about whether 2 species relationships are interlinked, how that predator-prey relationship works. We can put forth hypotheses, develop predictions from those hypotheses, and test those using mathematical models. This type of work is important and it's useful, particularly here, well, everywhere, but there are examples very close to home. This is the Atlantic cod. And this is the catch of Atlantic cod in Newfoundland, preceding and including the population bust in the late 1980s. And what we can observe here, so this is a fishery return. And you can see that the fishery increased slowly but steadily up to around about 1960. By the early 1980s, the population, or the yield from the fishery, the amount of fish being caught, increased dramatically. And eventually that hit a point where the population became unsustainable. We can think of here of humans as the lynx and cod as the hare. We got to a point where the humans were taking so many of the hares that the hare population collapsed. Had the fishery managers listened to the ecologists who were advising them that at this point the population was reaching that critical threshold, then potentially this could have been averted. And this is an ongoing debate that still runs between fishery in Canada, fishery in Northern Europe, where I'm from. But as we generate more information, we can feed it directly into these conservation programs. One final example before we wrap up. Anybody know what this is? Don't read it over there. Spruce budworm. Has anybody heard of spruce budworm? Yeah, one or two. Excellent. So spruce budworm are this little worm, little caterpillar, actually from moth, and they love to eat the buds on spruce trees. And their populations explode, typically on maybe a 30 to 40 year cycle, and they can decimate spruce forests simply by feeding on the fresh growth in the spruce trees. And at the moment, there's a population that's booming in southern Quebec, and there's an overspill from that starting to move down into New Brunswick. And there's a huge research project at the moment underway in the province to try and understand, one, what's going on with this booming budworm population, can we control it, manage it, mitigate it. And I'm going to leave it there for today. We'll pick up on this.